Hi, everybody. Thanks for the opportunity. Thank you to the organizers and the sponsors. So we'll take a small break, a small shift. I'm going to move this down a little bit. Is that okay still? All right. We're going to move away from the hippocampus to look at the work that we've been doing in the olfactory cortex. It's also an allocortical area. I like to think of it somewhat like the hippocampus stuck on the outside of the brain. So the goal of sensory systems is not merely to detect stimuli in the environment, but rather to extract the relevant salient features of those stimuli and represent them in a way that's adaptive and meaningful in order to drive, in this case, odor-driven behaviors. So if we think of the challenges that this dog needs to overcome in order to follow a scent to its source, one gets a sense of some of the complexity involved. So, for example, she needs to be exquisitely sensitive to a particular odor and be able to discriminate between two odors that may be very similar, right? At the same time, she needs to be able to generalize for the target odor and, for example, identify that odor over variable conditions such as an increase in odorant concentration. And so my lab is interested in trying to understand how these computations are affected in the brain. So odors are first detected when odorant molecules bind to olfactory sensory neurons in the nasal epithelium. Each sensory neuron expresses just one out of 1,000 different types of odorant receptor genes, and all sensory neurons expressing a given gene converge on this unique pair of structures in the olfactory bulb, the first stage of odor processing in the brain, called glomeruli. Right, so here you can see a series of neurons all expressing this one receptor, the P2 receptor, and the axons of these neurons all converge on the structure over there. The output cells of the olfactory bulb are the mitral and tufted cells, and these cells extend a single apical dendrite into just one glomerulus and project this information to downstream cortical olfactory areas. So the flow of odor information from the receptor through the sensory neuron into the glomerulus and into the cortex is essentially a labeled line that is specific for the particular molecular features of the odor. Consequently, if one were to image the activity of the mitral cell, and that's what Tomboza has done here, one can see that different odors will activate particular combinations of glomeruli or mitral cells that represent the molecular features of that odor. That information, the sort of elemental combination of elements, needs to be combined downstream of the olfactory bulb to form a synthetic representation of odor identity. And so that happens in the olfactory cortical areas, the largest of which is the primary olfactory cortex, or the piriform cortex. And so if one were to image activity of a population of neurons in piriform cortex, as we in my collaborator Alex Fleischman has done, one can see that here each of these dots represents an identified neuron in an anesthetized mouse, and the color code indicates neurons that are responsive to one of these three odors, and cells that are shown in white are responsive to multiple different odors. And so we can see that a given odor will activate this distributed ensembles of neurons in piriform cortex, and it could be said that particular ensemble, in fact, we and Alex have shown that the ensemble of neurons is sufficient to accurately represent the identity of the odor. Now, for the purposes of my talk, it will be very useful to understand a little bit about the structure of piriform cortex. As I mentioned, it's an allocortex. It's a three-layered cortex that contains essentially a single layer of principal neurons, or pyramidal cells, that are located in layer 2. These cells extend apical dendrites up towards the peel surface, where they receive excitatory synaptic connections from combinations of olfactory bulb mitral cells. The projection from bulb to cortex shows no topographic organization. Some people have said it's random. As far as we can tell, there's no evidence that it's not. That allows individual neurons within piriform cortex to respond to possibly unique combinations of activated glomeruli. In addition to their excitatory connections onto the pyramidal cells, these cells also make the mitral cells also make excitatory connections onto onto a class of inhibitory neurons located in layer 1 that provide feed-forward inhibition onto the pyramidal cells. Now, much like CA3, piriform cortex is a densely interconnected recurrent structure. in which excitatory axons from these pyramidal cells project across the extent of piriform cortex. This has the structure of an auto-associative network, similar to the types of things that we heard about from Jim and others this morning. And this recurrent circuitry is thought to be very important for things like pattern completion and amplification of specific types of odor receptors. I'll give the punchline away right now in case anybody needs to leave. I'm going to make the argument that that is only one and perhaps a small role of this extensive recurrent circuitry, because in addition to forming excitatory connections onto pyramidal cells, they also form very strong connections onto a separate class of interneurons located in layer 3 that provide very strong feedback inhibition onto the pyramidals, back onto the pyramidal cells. So let's return to the bulb and consider the following problem. I mentioned that a given odor will activate a specific combination of glomeruli, and one could presumably in some sort of cartoon model decode the identity of the odor from the specific combination of activated glomeruli. Right? But what happens if we change the concentration of the odor? We know perceptually that the quality or the identity of an odor is robust to large changes in concentration. However, the particular subset of activated glomeruli is quite steeply concentration dependent. This makes sense, right? Because these receptors are activated by mass action kinetics. So as you increase the concentration of the odor, so you'll increase the activation of receptors, and consequently here you can see, for example, for one odor over a range of one order of magnitude of concentration, there's a drastic increase in the number of activated glomeruli.
But it looks also like some glomeruli actually turn off as a...
Yeah, actually, they don't. One of the tricks that they did here is normalize everything to the highest point. So some glomeruli become more responsive at higher concentrations, but in fact...
But it's a nonlinear behavior.
That's correct. Right? So how do we solve this problem? And I think Lorenz is pointing to the fact that there may be fairly complicated solutions to this, but one relatively simple and elegant solution was proposed initially by John Hopfield. And he suggested the model is essentially this. Consider we have an odor A, that activates 3 glomeruli, indicated by the blue, green, and magenta circles and their constituent mitral cells. And this odor A activates these 3 glomeruli with a specific set of affinities, such that the blue glomerulus and mitral cells are most sensitive to that odor, and the magenta ones are least sensitive to that odor. Consider another related odor B, that activates the same subset of glomeruli, but with the reverse set of affinities. Hopfield's insight was that one might be able to separate these representations using time, and time related to the respiration cycle. So, for example, rodents or mammals breathe, and we have these respiration cycles that are typically on the order of a theta cycle, 300 or so milliseconds long. And so the insight that Hopfield had was the following. The most sensitive neurons, the most sensitive glomeruli, the most sensitive mitral cells actually, would be the ones that are activated earliest after inhalation. So in the case of odor A, the mitral cells associated with this blue glomerulus would fire before those associated with the green or the magenta glomeruli. Whereas for odor B, even though the same set of mitral cells are activated, in this case the magenta ones would fire earlier than the blue ones. And so if there was some way of sort of amplifying the impact of the early inputs, one could sort of avoid the spurious recognitions, using Hopfield's terminology, associated with the activation of these lower affinity glomeruli, particularly at higher concentrations.
Short question now. You've made now the transition from a rate code to a time code.
Yes, that's correct.
It's sort of a silent transition, because your images, they were not phase locked or anything. They were just rates. Right?
Yeah, we're going to move very quickly. I will not show another image for exactly that reason. Okay? The same model can be used for trying to generalize for concentration. So imagine now we just have odor A at either a low or a high concentration. Whoops, excuse me. At low concentration, only the high affinity glomeruli and mitral cells might be activated, perhaps with some weak activation of this moderate affinity glomerulus. At higher concentrations, all three of these glomeruli will be activated, but you'll always activate the most sensitive glomeruli and mitral cells before you activate the less sensitive ones, right?
I'm just objecting to just from where you are in your story to the word spurious, as if it's spurious information, but it clearly is part of the code of the odor, which of the glomeruli are all activated in which order. And so I'm just kind of curious why you're inclined to throw out the later activated glomeruli as if they don't carry information.
That is a contentious, I'm going to answer this in a few different ways. One, that's a contentious statement, and I don't necessarily agree with it. Two, Hopfield used the word spurious, and I'm jumping on it. Three, I would love to have this conversation, but it's going to take a while. So we can return to that maybe in the discussion. All right, so if one can use the same sort of temporal filtering strategy where one selectively responds to the earliest inputs and sort of ignores the stuff that comes later, then you can sort of solve this problem of concentration invariance, right? And a key prediction, and I should mention that there's now considerable evidence that odors are encoded using this type of temporal structure in the olfactory bulb. What's not known, and what is key for this model, is that the downstream decoder, i.e. the piriform cortex, should be selectively responsible to the earliest inputs that are coming from the bulb and should try and avoid, let me use it again, these spurious inputs that are coming later, right? So we wanted to ask whether or not that's the case. This is the first time I've tried to play this video. Let's see if it works. It should just run in the background. So to do that, we developed a method, particularly because these images are static, in which we can record from populations of neurons in awake-behaving animals. And to do this, we use 32-channel neuronexus probes that we lower either into the mitral cell layer in the olfactory bulb, or here into layer 2 of piriform cortex. And our electrode spans spans layer 2, and we can typically record from on the order of 100 neurons simultaneously. So here what I'm showing you is a trace in real time, where on the top we're measuring the respiration of the animal with a flow meter placed outside the nose, and on the bottom are the raster plots from 40 simultaneously recorded neurons. We trigger odor on exhalation, which is indicated here in gray, and measure the spikes within the first sniff after starting with inhalation. And people have shown, now, a number of groups have shown that animals can reliably discriminate odors within a single sniff, and we therefore realize that all the information that we need should be encoded within that sniff. And so today I'm going to ask three simple questions. I'm going to ask, how is odor information transformed from bulb to cortex? I'm going to ask, what are the circuit mechanisms that implement this transformation? And I'm going to ask, what are the computational consequences or the computational roles that each of these circuit mechanisms play? Okay, so here's an example of one experiment. Here we're recording simultaneously from mitral cells in the bulb and piriform neurons in the cortex. And here are raster plots from 20 mitral cell units and 68 piriform neurons. The red line indicates the beginning of inhalation. And what I've done is sort these neurons by their onset. And what you can see, so this is a single trial. This is the average over a number of firing rates. And what you can see is that what we find, which is in agreement with what others find, is that individual mitral cells respond throughout the sniff, and their onset latencies tile basically the extent of the sniff. By contrast, what we see in the piriform cortex is there's something quite different. There's a robust, there are a number of neurons that respond early in the sniff, and there's very little activity that happens later on. We can see that more clearly here. Now I've presented a single odor at multiple concentrations. Here a representative single neuron in either the bulb or the cortex in response to 3 different concentrations of hexanol. And what I'm showing you here, and you'll see a lot of these traces over the course of this talk, are population responses in which we record from individual units, we calculate peristimulus time histograms for each unit, and then we basically throw away all the single unit information and combine these to form a population firing rate. And so what you can see is that at the beginning of the sniff, there's a robust response in both the bulb and the cortex, but then later on, there's this large envelope of activity, which is actually quite concentration dependent in the bulb, that's just simply not represented in the cortex. It's like the cortex is sort of actively ignoring this stuff that's coming in later. Again, remember, these mitral cells are the direct afferent inputs to the piriform cortex. We used an optogenetic approach to go after this transformation a little more directly. In this case, we used a mouse line, a Thi1 channel-rhodopsin mouse line that expresses channel-rhodopsin in mitral cells, and recorded now just for sort of proof of principle from its single neuron on the dorsal surface. And we give 1 second long light pulses of different intensities, and what you can see is that there's this grained and sustained activation of mitral cells when we optogenetically activate the bulb. So now what we do is record simultaneously from these mice, recording now on the ventral surface of the bulb, so we're getting a lower bound estimate of the total output of the bulb, as well as in piriform cortex. And we give these 1 second long light pulses of different intensities, and what we find at the lowest intensity is that there's a small and sustained increase in firing in the bulb that's just not represented in the cortex. So perhaps we just weren't stimulating strongly enough, so we turned the laser on a little bit, we turn the laser power up a little bit, and now what we see is, again, there's a peak with a sustained phase in the cortex. In the bulb, that's just not represented. There's this transient response in the bulb, in the cortex. And as we turn the laser strength up more and more and more, we see the same thing. This rapid onset in cortex that's immediately suppressed to, or in fact even below baseline firing, and then that recovers quickly after the light goes off. So this indicates that the piriform is actively suppressing its responses to the later bulb input. What's the source of this suppression? I mentioned that there are these feed-forward intereurons that receive input from the mitral cells, and these seem to be well poised to shut off all but the earliest inputs. On the other hand, we have we think, based on slice work that I and others have done, that in fact it's this recurrent circuitry driving this feedback recurrent inhibition that seems to be, that is poised, or that may have a much more pronounced impact on the cortical response. So to address this, we again use an optogenetic approach, this time using a VGAT channel-rhodopsin mouse, in which we express channel-rhodopsin in all inhibitory interneurons. And we take advantage of the laminar segregation of the feed-forward interneurons in layer 1 and the feedback interneurons in layer 2 to be able to target which neurons we're recording from. So we lower our probe so that it spans layer 2, and if we turn the light on, because we're activating interneurons, we can reliably activate neurons in either layer 3 or layer 1 while suppressing the vast majority of VGAT negative pyramidal cells in layer 2. And we can then look at the odor responses in these different classes of interneurons. And what we find is that when we look at layer 1, we see that they have a fairly high firing rate, which corresponds to the reasonably high firing rate of the mitral cells. And so they're tonically active without a dramatic odor-evoked response. This suggests to us that this feed-forward inhibition is probably playing a role as some sort of high-pass filter for olfactory bulb input, but is not playing a pronounced role in shaping the cortical response. By contrast, if we look at the response of these feedback inhibitory neurons, we see that their activity is predicted by this brief increase in activity in the pyramidal cells, and their activity in turn predicts the pronounced suppression that we see in the pyramidal cells over the course of the sniff, indicating that they are likely responsible for shutting off, or that they are responsible for shutting off the cortical response. Now, because these feedback inhibitory neurons don't receive input from the mitral cells, but are driven instead by the pyramidal cells within the cortex, this indicates that the cortex itself is deciding what information is represented and what information is suppressed. And that points to a role for this recurrent collateral network in essentially implementing this temporal filter. So we wanted to test that, and to do that, we used the following strategy in which we can mute these pyramidal cells. We used an EMX Cre mouse that expresses Cre recombinase in pyramidal cells and unilaterally injected over the extensive piriform cortex in AAV that selectively expresses tetanus toxin in Cre-expressing neurons. This allows this will essentially block output from these pyramidal cells while allowing them to spike and leaving the rest of the circuit intact. And we've done a number of extensive controls in vitro to show that this is the case. In the interest of time today, I won't talk about those today. What we did now is record bilaterally from these unilaterally injected mice. And the first experiment I'll show you is what happens when we cross one of these mice with one of the Phi 1 mice. We can see that when we activate the bulb, we now see this large and sustained increase in firing on both hemispheres. However, when we look in the piriform, what we see in the control side is what we saw before, this rapid transient input followed by the sustained suppression. Whereas now, when we get rid of recurrent excitation, we now see the step function is transferred to the cortex, or is represented in the cortex. What happens when we look at odor? This is the population response from the contralateral side. You can see, again, this very brief blip followed by a pronounced suppression. I want to point out that an individual neuron receives on the order of 10 times more recurrent inputs than it does afferent inputs. And so it was somewhat remarkable to us that when we got rid of the recurrent excitation, the vast majority of the excitatory input onto these cells, instead of seeing a role for this recurrent network in amplifying the response, which is what one might expect, we saw that that the responses were in fact much, much larger when recurrent inhibition was relieved. We also looked at the responses to different odorants, and here I'm just showing total population firing rate as a function of odor concentration. And what we saw before is that under normal conditions, there's a robust normalization of cortical output across odorant concentrations. And now what we see is that the cortex becomes very steeply concentration dependent. Finally, we looked at odor coding. What I'm showing you here is the accuracy of a simple linear classifier as a function of number of cells to responses under 3 different conditions. Responses to 6 different odorants at a given nominal concentration. Decoding responses to 6 different odorants at a nominal concentration. Decoding responses to a single odorant at multiple concentrations, or generalizing for odorant identity across a range of concentrations, effectively asking the decoder to identify a familiar odor at a novel concentration. And what we can see is that in the control case, the decoder does relatively well. However, in the tetanus case, we see effects that are rather interesting. There's a small decrease in the ability to classify the different odorants, which would be sort of consistent with the idea that this recurrent circuitry helps with pattern separation or decorrelation of these overlapping inputs. There's no real effect on concentration decoding, which makes sense given that these recurrent circuits are helping to normalize the response. But there's a pronounced loss in the ability to generalize for odorant identity across concentration. So what do we think is happening here? Under normal conditions, this is what I showed you earlier. Let's imagine these 2 odors, odor A and odor B, that have overlapping inputs. What happens effectively in the cortex is that what you're representing are the earliest inputs, and this allows you to decorrelate these inputs that are somewhat overlapping. In the case of different concentrations, by responding selectively to the earliest inputs, oh, I messed that up. That should be blue on the bottom. Sorry, I made this last night. What happens here is that you're just representing the same odor. That's a bit of a slip. All right, and so let me quickly summarize. And when we eliminate the recurrent circuitry and we basically are now driving the cortex just by bulb input, what happens is that these correlated inputs result in sort of confusing patterns in the cortex, and this concentration and the ability to sort of form a concentration-invariant representation of the odor identity is corrupted. Let me summarize by saying that the earliest bulb inputs seem to primarily define the cortical odor representation, that this is implemented by some sort of winner-take-all, temporal winner-take-all filtering that's implemented by this long-range recurrent circuitry driving strong feedback inhibition. And this supports concentration-invariant odor recognition in the cortex. And with that, I will thank you for your attention. I'd also like to thank the people who did the work. Most of this work has been done by a postdoc in my lab, Kevin Boulding. Work that I haven't spoken about from my collaborators and my funding sources. And thank you for your attention.
Questions? Peter.
There are 3 quick questions. You probably answered this. Are these awake or anesthetized animals?
Awake. They're awake behaving animals.
And you tried both familiar and novel odors?
We've only tried What do you mean familiar and novel odors?
Familiar, it's a little vague. And odors the animals have sort of learned.
No. In some cases, we recorded multiple times from a given animal. Typically no more than 2 or 3 recordings per animal, and we're using sort of standard monomolecular sigma odorants. So there's no prior experience with these
Third one
odors.
is the code presenting multiple odors?
No, not yet.
Question. Yeah.
Could you clarify what you mean by concentration-invariant coding? Because it seems like you can read out both the identity and the concentration from the code, right?
Yes.
So is what you're saying closer to saying that you encode both with
You're encoding both.
but with an overall constant firing rate or something?
Yeah, so we published a paper earlier, I guess, well, not this year, last year in eLife, where we sort of go into this in quite a lot of detail, and I'd encourage you to look at that. But basically what we find is sort of complementary, almost multiplex coding schemes for identity and intensity. So odor identity is encoded by the specific combination of activated neurons in piriform cortex over the course of a sniff. And the intensity, or the concentration, seems to be encoded using some sort of temporal coding structure in which intensity is encoded by the synchrony of the ensemble. So what I'm talking about here by concentration-invariant coding is the ability to generalize for identity over a range of concentrations. So I'm talking about, I should say, concentration-invariant odor recognition is a better way to put that.
Farther. Kevin, I brought this up a little earlier. We're talking about what you lose when you chop off the later activated glomerulus.
Yep.
Because it seems like you're saying, kind of to a first approximation, the first glomerulus that's active becomes the code, because the later ones...
The first glomeruli that are active.
Right, but I was kind of wondering about that, because if it was literally that the first input, then thereafter, in other words, the first activated glomerulus is active and it sort of shuts off the cortex after that,
Yeah.
then there'd only be 1,000 different odors that you could recognize, because there's only 1,000.
Yeah, no, but what I'm saying is that the first input doesn't need to be 1. It could be 4 or 5 or 7 out of 1,000.
Okay.
Okay? And I think evolutionarily this makes sense. You have a large number of molecules in the environment that you need to be able to detect. And be able to detect at very low concentrations. So one solution that one might use to solve this problem is you create an array of 1,000 different detectors that are all exquisitely sensitive to different features. And sort of the cost of doing business is that those detectors, those receptors that are exquisitely sensitive to a small subset of the possible number of odors that exist are going to also respond at high concentration of other inputs, right? The way to select for the ones that are sort of most optimally tuned for this particular odor would be to look at the ones that are strongly activated at the very, very beginning of the sniff.
So I was wondering how this generalizes to multiple odors, or to what extent animals are actually able to detect a mixture of multiple odors, because then sort of the combination might screw up somehow.
Absolutely. So this, Peter asked if we've done mixtures. We have, but not very well. My supervisor's lab. My supervisor has done this, my previous supervisor has done this, and what they found is pronounced mixture suppression. There's not a lot of quantification in that. But basically what they saw, this is a paper from Stetler and Axel in 2009, is that a given odor A will activate on the order of 10% of the neurons in piriform cortex. Odor B will activate approximately 10% of the neurons in piriform cortex. And odor A plus B together will activate approximately 10% of the neurons in piriform cortex, where that mixed combination is largely a subset of some of the neurons that were activated by odor A and some of the neurons that were activated by odor B, with a few new ones and many of the ones that were activated by either A or B dropping out. They have no idea what underlies that mixture suppression. I think that our model solves this, but we don't have data for that yet. I think it solves it in the following way. I'm not, again, sort of speaking to Bartlett's question, I'm not suggesting that it's this first glomerulus that defines the odor response. There are 1,000 different glomeruli, and we can tile the sniff with all 1,000. Let's imagine it's more sparse. Let's imagine we tile the sniff with all 10, with 100, right? And let's suppose that the first 10 are the ones that define the odor response. So you'll have a rank order, a latency order of each of the activated mitral cells for odor A and odor B, and basically if you combine these, then what you could imagine is that the response to the mixture of those most strongly activated by A and B, right? We haven't done that. I think that's what's going on. And I think what happens with the mixture is you get this sort of the most robust elements of that mixture.
Then you need
But I don't
still a readout mechanism for this overlay superposition, sort of to see that these are two different odors, like in hyperdimensional computing or something. Okay.
Unless the percept that emerges when you put these mixtures together changes.
Yeah, it could be.
Right?
That's
Synthetically.
a behavioral question. Are animals actually, I mean, I suspect they are able to say, well, this is a mixture of strawberry and vanilla or something.
Yeah, so there's been some psychophysical work in humans with this, and it's I think we can take it with a grain of salt. A trained perfumer or a sommelier can pick out notes in a perfume or a wine, right? An untrained person smelling novel combinations of odors typically has a lot of difficulty identifying the elements in a mixture of more than 3 or 4 components. At that point, it basically becomes a synthetic sort of gestalt representation of the mixture, and the individual components become indistinguishable. So that needs to be done more and better, but that's what we think we see.
Just one last question.
Do you think there's an advantage to the system doing this winner-take-all network at the cortex level? It seems like you could do it earlier and not bother with transmitting all of those extra spikes. Like have glomeruli inhibit each other.
So one advantage might be the following. I mentioned the piriform cortex is so this is speculation, right? The piriform cortex is the largest of the cortical olfactory areas, but it's not the exclusive target of the olfactory bulb mitral cell, of the olfactory bulb. It's the cortical structure where associative odor representations are thought to be formed, right? It's where we think we form the identity of odors that have no intrinsic or biologically innate importance. And so that seems like it might be a good place in which to implement this filter. However, there are odorants that are biologically or that are innately relevant. So for example, in the case of a mouse, that would be fox urine. Right? So you could imagine a low concentration of fox urine, and we think, we being the field, that mitral cells project to a number of different cortical targets. The largest of which is piriform cortex. They also project to cortical amygdala, the tubercle, and lateral entorhinal cortex. It's thought that the piriform cortex is where these associative representations of odor are formed, whereas innately relevant odors seem to project to the amygdala, and that might be driving innate responses. Okay, so in that case, you might not want to filter the stuff upstream. You might want to let that get through. And if you have low concentrations of fox urine over a background of something that's very, very, very high, then if you implement the temporal filter upstream, you might lose that. That's how we're
All right, well, we should probably move on. Thanks very much.